Welcome to the Proteomics in Proximity podcast, where your co-hosts Cindy Lawley and Sarantis Chlamydas from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Cindy and Sarantis.
Hello, everyone. Thanks so much for joining Proteomics in Proximity. I'm one of your co-hosts, Cindy Lawley, and I have with me my other co-host.
Hello, everybody. I'm Sarantis. I'm happy to be here.
Very good. So today we are joined by Koprulu and Claudia Langenberg. We are talking today about a wonderful paper that came out in Nature Metabolism in March called Proteogenomic Links to Human Metabolic Diseases. Very exciting paper with, by Claudia Langenberg and Koprulu standards, a modest sample size, but amazing findings. So I think it's really enabling. And we'll dig into that. But first, let's introduce our guests. Sarantis, do you want to do the honors?
Yeah, absolutely. I'm start with Mina. Mina is a PhD student from Gates Cambridge Scholarship and supervised by Dr. Claudia Langenberg. Before her PhD, he did her Bachelor of Studies in Human Genetics at UCL, and then separately she completes her Bachelor of Philosophy in Genomic Medicine at the University of Cambridge, working in genomics data and genomics data and dealing with population studies and the UK biobank data sets. And we are really looking forward to hear about your project and paper Proteogenomics.
Yeah, and digging into how you got where you are. Anything you want to add to that, Mine, that you want people to know going in?
No, I think that's a very fair introduction, and it's lovely to be here. And I'm looking forward to chatting a bit more about both our journey and our recent publication.
Awesome. Fantastic. So then I have the privilege and the opportunity to introduce Claudia Langenberg. Now, Claudia is probably someone who needs no introduction, but I think the impact that she's had in the publication space is huge. Last I saw, she had, I think this was in 2021, over 300 peer-reviewed publications at her very young age, and I think over 40,000 citations at that point, and that's already several years old. So this is a world-renowned scholar that is working with Mine. And of course, many of her publications are in the journal series like Nature, JAMA, Lancet, very prestigious and very impactful, working with massive data sets. So I think of her as a large-scale population epidemiologist, such a big word, but it integrates many of the omics. And I think she keeps an eye on the technology advances to be able to build systematic approaches to understanding human biology. So Claudia, I think now you're at Queen Mary, University of London. I think when I met you originally, you were at the Berlin Institute of Health and Charité. So I'd love for you to just tell us a little bit about your position now and your affiliations, if you don't mind.
Well, thank you so much for the extremely kind introduction. And I think the most complimentary thing was about the young age, which is not only a compliment, but a lie. So this everything puts everything in perspective. And yes, also I should say the work that we've done, and Mina is a student at the University of Cambridge, where I've had a dual affiliation and a long career prior to me arriving at the Berlin Institute of Health. So originally being German and training as a doctor in Germany, I came to the UK, and I was in London originally, where I did my PhD in epidemiology and a master's at the London School of Hygiene. And before I then went to Cambridge to focus more specifically on genomics, and then later other omics. And so, yeah, it's a huge privilege to be here, and it's an even bigger privilege to work with talented people like Mina and others in our team. So it's a real team effort and a lot of fun to do that and work with people like that.
It does seem like you have fun. And I'll tell you what, whenever I've talked to Mine and when I've seen her present, she's always got a big smile on her face. So I would love to dig into your journey to end up in Claudia's lab. Mine, do you mind giving us a little background and how you got interested in science? I mean, feel free to start wherever you want.
Yeah, sure. I've always been very interested in science and especially biology, but especially I always wanted to do something with my career which improves the lives of others, so contributes to the society in a meaningful way. And as I said, given I was quite interested in biology, human genetics especially quite interested me because a lot of the sciences are quite established in the sense that, human genome is very recent compared to many other scientific fields, and there's so much unknown. And I think that is especially what interested me, as well as its potential to, improve the current healthcare system and the clinical translational potential of the human genetics findings. So that was my inspiration to go into that. And unlike some other people, I think I've been in a quite narrow, specific path ever since then. So I did my bachelor's at UCL, as Sarantis mentioned, in human genetics specifically. And after that, I did my master's in University of Cambridge in genomic medicine, which is where I got introduced to bioinformatics. So I did my master's project at Sanger Institute, working on UK Biobank, as well as whole genome sequencing data for population isolates, with Elisa Gini's lab at the time. And after that, I moved back home, which is Turkey for me, to work on rare disease genetics. So I spent 2 years back in Turkey at Boğaziçi University, researching rare disease genetics. So that was more studying whole exome sequencing data from consanguineous marriages or consanguineous parents having sort of disabled children, trying to better pinpoint the exact genetic changes that cause such severe rare disorders. And in the meantime, while I was back home, I was thinking about my PhD and what I might want to pursue, and multi-omic integration, so better understanding the different omic layers and how that can actually improve our understanding of genomic studies and their clinical translation was what's interested me. So I got in touch with Claudia and we had a brief interview, and that was, I think, a beginning of such a beautiful collaboration, at least on my part.
And then starting a little bit about your paper that you have presently, would you like to explain a little bit about this Epic Norfolk study, this Epic Norfolk cohort? What's the characteristics of this cohort and why it makes it so special?
Sure. Epic Norfolk study is a population cohort that was first established in the beginning of 1990. So it has approximately 30,000 participants that were followed up phenotypically. So in terms of different behavioral and sort of healthcare characteristics for several years, as well as linkage to their health records. And we had proteomic samples measured from 3,000 of Epic Norfolk participants. But Claudia, if you have anything more to add on Epic Norfolk study, please.
No, I think I can say one of the opportunities this study has offered, and I think the beauty of such prospective cohorts such as Epic Norfolk and UK Biobank and so on, things setting up in the past, is we benefit from the samples that were stored at baseline in liquid nitrogen. And then those samples are so valid because anything you can do in the future, now you measure 3,000, then you measure 5,000, then you measure 10 and more 1,000 proteins. So anything we use now, we won't be able to use in the future. So we're always very cautious of that, conscious of that, and hence cautious with our sample use. So it's really important that proteomic technologies now use such little sample. So you could have used more sample in the past to measure 3 proteins. So to be able to use thousands in such a small amount of blood is absolutely amazing. And to have the opportunity, we weren't PIs, we're not PIs of those cohorts, the foresight of setting this up, Nick Wareham in Cambridge, and before that, Katie Core and others who've led this study, and other cohorts that we had the privilege of using the samples for. It's really, really amazing to be able to do that. That's one thing. But the other thing that's important is as time elapses, these codes become even more valuable because, sadly, people have events, they develop diseases, they die. So if you then look backwards, because you have samples stored, provides the opportunity to have such an efficient design of doing it. You don't have to measure everybody, but you can have a kind of what I call a very nifty design in choosing people who have developed a disease, i.e. it's new onset disease, but the sample was stored before they had it. So you avoid this kind of reverse causation by which the disease impacts your proteome, and hence you can't really disassociate this. So this is what we exactly did in this context. And Mina's study was one of the projects that we did on the context of that design. But you can also look at different diseases based on the people who developed them in the study, use that baseline sample, and then you use a big control cohort of people who serve as the controls for many different diseases. It's called a nested case cohort study. And it's a really beautiful design, but that can only be used if you had the foresight of setting up a large prospective cohort like that. So we're very grateful to all the participants of Epic Notebook and the PIs, of course, who've enabled the use of that. And also coming to the cost of some of these kind of very, informative molecular technologies such as yours is, of course, they're not cheap. And hence that means if you can use a design that minimizes the number of samples you have to use for a given purpose, of course, that's incredibly useful for us. So,
and I think I just wanted to comment on this liquid nitrogen aspect, this ability. I think this foresight. I work with a lot of groups that do a lot of different things with large population health studies. There are very few cohorts that had that much attention to reducing pre-analytical variation and tracking it as is documented in this cohort. So that's exciting to see. I think it's maybe less important for proteomics than metabolomics as an example, but you work in all of that. And so, yeah, I just wanted to underscore that point. Yeah, Sarantis, please go ahead. Okay.
Would you like, Nina, to summarize a little bit the take-home messages of the main take-home of your paper and what's so exciting on this paper?
Yeah, of course. And just also clarify, the paper, I was the first author, but of course it was a teamwork and everyone contributed a lot making this work possible, including the Epic Norfolk participants, of course. Just to summarize the paper, I think I could just briefly say we looked at the sort of systematically linking genetic variants, blood protein levels, as well as disease risk data to be able to pinpoint causal genes and proteins that underlie diseases. So just to give a bit of background, since enablement of the genotyping technology, a lot of studies have been conducted associating genetic variants with different disease risks or disease susceptibility. And today, 200,000 genetic variant disease associations have been established and are publicly available. Looking at such numbers and figures, we would have assumed we know the biological basis for all diseases. However, we all know that's not at all the case. And I think that's where proteomics and different sort of omic layers come into place in helping us better understand the disease mechanisms, actually what's happening underlying the diseases in our body. So in this study, we had samples from 3,000 individuals measuring approximately 3,000 blood protein levels. So we first looked at the genetic regulation in the cis regions. So the cis regions are the protein encoding regions and sort of flanking regions around the gene for the protein target itself because of our moderately sample size, as we've already mentioned. So we looked at the cis genetic regulation for different blood protein levels, some of which had never been targeted before, given the recent Olink platform. And then we have used that knowledge, that sort of proteogenomic knowledge, to better understand causal genes of proteins that underlie diseases in a systematic manner. So we have first looked at shared genetic regulation for different disease outcomes and blood protein level regulation, PKTLs as we call it, protein quantitative trait loci. And we identified 224 targets that regulate approximately 500 different traits. And we also refined the causal genes or proteins for 40% of the previously established genetic risk loci, which sort of highlighted that even moderately sized proteogenomic studies can contribute to uncover novel biology for existing risk loci that were published in the literature. And finally, we looked at the convergence of the PTTL studies and the regulation with the rare variant gene burden analysis. So on one hand, comparing the loss of function of the genes with the sort of genetic regulation of the proteins. And
So I wanted to click back on this, the disease associations that were enabled by genetics. I think the ability to make those associations in small sample sizes was like low-hanging fruit in the early days of that sort of GWAS era, which I think of maybe early 2000s, right? 2005, 2006. That's when those things started really ramping up discoveries. And then they became harder and harder to make those associations as those really strong associations were discovered and documented, and then larger and larger populations were needed to make those associations. I think what you're saying is that these other layers are helping you to do more with those more modest populations. And Claudia already made the point about the costs of layering on metabolomics or proteomics or these additional omics. Can you say something about what that enables and what you think will happen in the future there?
Yeah, of course. So basically, as I mentioned, and as you mentioned already, thousands of genetic variant to disease associations have been made. And those were very valuable and contributed to our understanding of diseases to some extent. However, majority of those associations fell into non-coding regions of the genome, meaning it was quite difficult to actually interpret which causal gene or protein was acting in causing the disease. Hence, it was quite difficult to understand the pathways and the mechanisms. And while we don't have a functional target, it's difficult to actually build more effective and safer therapies or repurpose existing ones. So in that sense, having the additional layer of the proteomics data actually helps us to pinpoint a functional entity that plays a role in the disease. As I said, there's certain statistical methods that help us better understand the shared genetic regulation of both the disease risk and the blood protein levels or abundance of certain proteins. And if we see strong statistical evidence for shared genetic regulation, then within a large region of many candidate genes, we can actually refine it to a single candidate gene or protein for particular diseases in a systematic way, which can then be used for, as I mentioned, intervention or more targeted therapies, safer therapies. So in that sense, even moderately sized molecular QTL studies can really help us better understand the disease and pathways that are involved, and also build more effective therapies.
I think we're on a path. I think this is a path for discovery that is, I think, just going to ramp up, I think, very similarly to what we've seen those discoveries enabled by genetics. I'm really excited about that. I always think about these different diseases that might share pathways in common, almost like a Venn diagram, and understanding the complexities of why certain proteins or protein pathways are related to, like, I don't know, caspase, I think, is like showing up in breast cancer and asthma it's an example I think of. And those diseases are so disparate to me. I wonder if you think that proteins or pathways that are showing up as causal, and I think you make a good point about why causality is so important for therapies. Do you expect those same proteins to be causal in other diseases? Like, what do you think about that? You're much more versed in seeing the data.
So I think that is sort of the beauty of our study design, that we sort of approach the data, different layers of biological data, starting from the genome all the way to the phenome in a hypothesis-free manner. So doing that and looking at the data systematically without any sort of prior hypothesis allows us to see what the data tells us. So as you mentioned, certainly looking at what we have discovered in our paper is that genetically anchored proteogenomic studies, so the proteomic studies, can help us discover molecular hubs of associations of proteins with diseases that we wouldn't have predicted otherwise from just the general literature or prior knowledge. And that is actually quite interesting to follow up because, the data uncovers something that was unexpected. But likewise, we can also see quite specific protein disease pairs, which can also allow us to have quite specific therapies for potentially not intervened diseases before. But just one more thing to add is that we currently have not a very full, complete picture of the proteomics. So we're only able to do that for the proteins we're able to target. So hopefully in the future, you have asked about the future directions before, having a more complete idea about the full landscape of proteomics would be ideal to better understand those molecular hubs that we have just talked about.
But to maybe I can add one small thing to this, because as Mina's work and other proteogenomic work that we've previously done in the group has shown, is exactly the beauty of that. As you scale up the number of proteins and as you scale up the number of diseases you can look at, this kind of proteogenomic approach that doesn't require that each of these layers is measured in the same sample size. You can utilize the power that you have across any biggest genomic study for a disease that you have, together with your protein genetic study. And that is the beauty of looking exactly like you say, Cindy, at the overlap of a specific gene to protein across any disease that you can look at. And because the community is sharing data openly, or the summary statistics for each of these diseases is shared openly, for most diseases, cancer is lagging behind, sadly, a little bit in terms of the openness. But for many diseases that is there, it enables us to do exactly that, to draw a whole map from gene to protein to disease. And is that map, is that link coincidental, or is it possibly causal and a real shared genetic signal? And that's such a good way of prioritizing what you then use for the experimental work downstream, because of course this is a computational approach and not an ultimate proof, but it's such a good and data-driven way of prioritizing links between diseases, links for where we have already, specific drug targets, new ones, potentially adverse effects. So it's a really versatile way of looking at all of that. And then just maybe to add one small thing again, as you said, is you can kind of ramp this up as you increase the number of proteins, or as you increase the density of your genomic array, or the coverage of rare variants and by sequencing and so on. But of course, an important part of really making this more useful is increasing the phenotypic spectrum. And that really is only possible if we move away from diseases we all study opportunistically to diseases that you can't really easily measure or nobody's interested in, but they're still really important for patients. And a lot less headway has been made in terms of understanding their genetics or releasing the summary statistics for those studies. And that's really where huge studies come in that have electronic health record data from GPs, from hospitals, from death certificates, and bring all of this together. And that's why, for example, UK Biobank, but also FinGen and many other endeavors around the world can really help us to not just increase the molecular side, but the phenotypic side. And that is so important to also link diseases which have not really been so much in the center of attention, but need to be.
Can you give an example of one of those diseases? Are we thinking rare disease here?
So I think it can be across the frequency spectrum. It can be across specialties. I think disease examples that I would choose are those that possibly are not as easily diagnosed, are not severe enough to always require hospitalization, because I think most people around the world have tried to get their hospitalization data ICD coded. It's relatively easy. But the data, for example, in the UK that comes from, or in England, from primary care records is harder to map. There's kind of a bit more diversity in terms of systems, the data structures and so on. So diseases that are predominantly managed and diagnosed in primary care are where we need to move towards.
Oh, that's so interesting. Yeah, I appreciate that point.
To technical question. You have some false positive, right? And then would you like to see how do you make the selection of false positive, and how you define this in your analysis compared to other technologies, for example, that you have tried before?
Sure. You're asking about false positives.
False positives, right? Yes, yes.
So in our analysis, we try to be quite careful, given we're working with data and diseases, as you mentioned. So in terms of our analysis, we always use quite a rigorous threshold to report what is statistically significant. So in terms of our statistical threshold, we always correct for genome-wide significance times the number of proteins that we're including in the study to sort of minimize the error, including thousands of targets in this case. So we usually go for quite a rigorous statistical threshold, as well as good QC before we feed in the data, of course.
So that's great. You mentioned before also that a lot of this falls in non-coding regions, right? A lot of these specific genes may fall in non-coding regions. Do you have an idea what type of these regions may are promoters? They are, I don't know, they are enhancers. Do you have an idea? Can you map these regions?
So we map all the variants in terms of what they're predicted to be in the genome. So we look at, the proportion of protein altering variants, those variants that fall into the protein encoding region itself and alter the shape or the structure of the variant, the protein, and the percentage that fall into non-coding regions. However, there's quite limited knowledge about the functional characterization of the non-coding variants themselves, apart from particular groups studying particular genes and more sort of cellular or functional models. So in terms of our work, so far we have only computationally annotated the predicted consequence of the variants. However, of course, further follow-up would enlighten what exactly those non-coding variants do in terms of cis-regulation of the protein itself.
That's great. And also, I think a lot of studies, and then from the literature, as you mentioned, for different proteins, from the literature, from some mice knockouts, you can have an idea of how they regulated and how good they regulated. Is there any plan to follow up more with mice or with more knockouts? Is there any plan in the future to follow some of these targets or collaborations that we are doing in this respect? If you can share that information.
I mean, we of course always welcome any sort of collaborations on the functional characterization. The idea of doing these studies is not to put a long Excel sheet as we have done so far in terms of our supplementary table, but rather genuinely understand the biological mechanisms that underlie diseases and how that can contribute to clinical translation of some of the findings in the sort of longer run. So with that, maybe an open call that if anyone finds any of the targets that we highlight interesting, we are, of course, very keen to functionally characterize and better understand the mechanisms.
I think in the paper you mentioned something about, I was just looking for the reference, something about cellular models and the history of demonstrating functional associations or an understanding of function around cellular models, and some of the how this approach can be a complementary way to add an understanding of function. Do you mind talking a little bit about that, like where this fits into our traditional approaches, certainly like where it might fit in understanding what we're learning from single cell or spatial work, that some of those technologies are really advancing right now? I'm curious your thoughts on those.
No, of course. So what we do in terms of our team is more bioinformatics. So what we work with is what the data and sort of predictions and better understanding the computational and statistical conclusions we can draw from the data. However, as we all know, and as you have highlighted, there are complementary ways in better understanding these. And basically, we are very interested to see whether what we observe computationally and statistically actually translates into biology, starting from cellular models, building up to animal models and sort of human biology. So what we observe in almost quite isolated, looking at singular sort of targets and singular genetic variants, we would be quite interested in whether our conclusions hold or what sort of variations from our conclusions we see in different models. So I definitely agree that they're complementary and there's ways of integrating those knowledge to build a more comprehensive or sort of holistic understanding of the biological mechanisms.
Oh, I love it. I love the background you have both in an understanding of epidemiology and your mad bioinformatics skills. Where does machine learning fit into all of this? Is it something Yeah, I'll just stop with the question.
No, of course. Machine learning and artificial intelligence is certainly fields that are very quickly developing and are grabbing a lot of attention at the moment. And some of the software we use are considered basic machine learning models themselves. But in terms of applying machine learning or artificial intelligence to sort of proteogenomic studies or more biological studies at the moment is rather challenging because, as we have all spoken about, this large, high throughput biological data is recently being generated. And they are so recent that we're spending a lot of time better understanding what they mean and what they actually are. And currently, the biological data, the way they are, actually violates a lot of the assumptions that machine learning and artificial intelligence models make. So I think in the future, they can certainly be very useful, but we certainly need to be cautious and better understanding our data before feeding them into the models, I would personally say.
And maybe I can add something to this. So I'm currently sitting in what's called the DERI. So when I came to QMUL, so Queen Mary University of London last September, so I had this opportunity to set up this new Institute for Precision Healthcare. And it's kind of quite unique because it's a cross-faculty institute. So it really draws in very broad range of expertise. And, it's a big plans and developments for new life sciences building. And that's all quite a way away. So we have to wait for this. So we need an interim space. And we're very lucky, and it's actually not coincidence, where I'm sitting right now is called the DERI. It's the only other cross-faculty institute that QMUL has, and DERI stands for the Digital Environment Research Institute, which is focused on AI. And so it's not just AI in healthcare, it's AI across a broad range of applications, from games. We're sitting on the second floor with the games people, which my kids think I have the best job in the world being with games people. They want actually what they don't know when they have conversations about board games. I've not heard of a single one of them. So I can't mention Monopoly or anything else. But anyway, having said that, this is the environment in which it can flourish. So we focus on health and on healthcare data, and DERI has that as part of its remit as well. So it's really important to bring this together. And, going away from the kind of, this pitch on how it's important, the concrete thing that we're already doing is, the molecular data is so complex. You need smart and efficient and unbiased ways of data reduction. And that at the moment is most of the use for machine learning in our arena of work. So where we kind of try to, let's say, predict diseases, how are you going to prioritize? Nobody wants to go into the clinic and measure 3,000 proteins to predict a disease. Do you even have any benefit of what's the incremental value of measuring proteins once you have a core set of 10 most important ones? So those are all questions that machine learning can help us to address more systematically, and that's incredibly useful. So it's a steep learning curve for us as well. It's not our bread and butter. We're not the methodological experts of developing it. But being in an institute like this one, for example, means that we're only very short way removed or not removed from the people who do develop these methods, and that we at the right time can employ them, can test, are they useful, could they be biased? And so that's really, really, it's a great opportunity, I think.
Well, I think those people that are developing those methods aren't going to have the biological know-how to know when they've overtrained those methods and are going off on a spurious tangent. So the context, I think, is essential. I think Mine makes that point.
Yeah, exactly. And it's this, it's a team effort, right? And you need the people who have the samples and the clinical knowledge. You need the people who have some of the kind of bioinformatics, computational, and you need the, methods developers. So it's beautiful. Nobody can do all of it themselves, alone. So I think it's a great.
It's CQ, right? It's collaboration quotient, right? You have your IQ, your EQ, and your CQ. And my experience is once you develop your collaboration quotient, which you're teaching your students and your postdocs and all of that, if they don't already have it, Claudia, once you have it, you can't go back, I don't think. I think once you experience the joy and the ability to work together cross-functionally, it's really remarkable what can be accomplished.
Well, coming back to the question that Sarantis was asking, and I think it's a very good one, and as Mina has said, so part of the problem that we have encountered is, here's Mina and comes up with these beautiful candidates. And it's almost, there's two things that are important to kind of what she said so well, which is using humans as the model organism is a great way of prioritization. That's number one. But the second one is kind of, if you come from a Bayesian framework, you really want to find out what's your greatest chance of success. How do you increase your prior of this having success? And given that the experimental follow-up, is expensive, it's lengthy, and can go wrong in so many ways, it is really a great opportunity. It's only, to prioritize on the basis of human proteogenomics, as Mina has said. The problem is, how do you get people who have the experimental setup to come to our results and take it? Because that's not as easy as we thought. We think, here's
I'm so excited to see it.
We go to the world expert in this and bringing them, our example. It's hard. It's hard to motivate people to step out of what they're already doing and focus on your finding. It's hard if you have somebody who's focused their whole life on a given pet protein to say, how about this one? And then we need help, I think, to try and learn how we engage the relevant clinicians and, molecular biologists and other people to also use our results rather than at least, complementing what they're already doing in order to be able to follow some of it up, because we do depend on the functional follow-up and on the expertise of those people. So how do we reach out? How can you help us to reach out to the relevant people to really make it worthwhile?
What are the titles of those people? I mean, what are their jobs? I know there's more than one, but I think of translational, right? That certainly translational scientists or implementation scientists, or maybe those are pieces of it. But who are the ones that we need to listen to this?
So I think it depends on the stage of translation that you're at. If you're really early for the experimental workup, it's a very different set of people than it would be for the people if you have a different kind of work where you really are ready to maybe possibly already consider an initial trial, or you need a relevant clinician or head of that clinical department to enable kind of the setup within the, let's say, hospital. So I think it really depends on what level you're talking. The initial level, I think, is for us the experimental follow-up and validation, because what we do is statistics and probability, but it's still an association. We're under no doubt. We do not prove causality. And so that kind of functional follow-up is crucial. And those are the people at the first stage.
And I think also you don't know the cause. Some of those cases, you don't know the cause or the effect, right? You don't know if this what you see in the protein levels is a cause or could be the effect, or could be, both PT, right? The last comment to me, I really like when you see the colocalization with EQTLs from different tissues. And this is also a question like what we see in the plasma proteome, it reflects what happens in the tissues. And do you have any comment on that? How do you see that this is correlated, and what is the distribution, the contribution of the singular tissue to the plasma proteome? Do you have some examples or some ideas on that?
And a shout out to Nomad and GTEx, I will say. Fantastic resources. Yes.
No, we are very grateful for all the publicly available resources, ranging from the EQTL studies all the way to the GWAS summary statistics, which have made our work possible as well. In terms of the EQTL overlap, that is rather challenging in our field, in the sense that we see, for example, in our study that only approximately half of the PQTLs that we see sort of are in close linkage disequilibrium with an EQTL. And in certain examples, we see beautiful overlap, right? Like we find an example with a disease that acts in a particular tissue, and we see colocalization, quite strong colocalization with the tissue of interest, where the story becomes quite beautiful and everything makes sense. However, there's also the flip of the coin, where we see in some examples quite limited overlap between the PQTL and the EQTL data. And although our current paper hasn't really focused on that, I think that is something that we as a community need to better understand, where there is overlap and where there's a lack of, and what might be the underlying reasons, definitely.
I think this is a great spot to mention the really interesting link to type 2 diabetes that you found in here that we want someone to follow up on, as Claudia mentions. Do you want to just summarize that really quickly, that pathway?
Yeah, of course. One of the examples that we have noticed or we have highlighted in our paper is the gastrin-releasing peptide, or GRP for short, and its linked with type 2 diabetes. So what we saw in our paper is that beautifully different layers of biological data, so evidence from mice studies, evidence from human data, as we have been talking about, and different sort of cellular models all overlapped with the same conclusion. So what we've identified was that the higher levels of GRP in human plasma were co-localizing with lower risk of type 2 diabetes. And when we integrated in the different sort of intermediate layers, which are different body fat distribution traits, so where we accumulate fat in our body as well as the overall fat, what we observed was that higher levels of GRP was leading to less fat accumulation overall in our body, leading to lower risk of type 2 diabetes. And as I mentioned, there was previous studies that were published where human recombinant GRP led to actually reduced food intake and weight loss. So we have highlighted in our paper that GRP can be a new example for a potential therapy for type 2 diabetes by decreasing lowering the fat accumulation in our body.
It's actually a hot topic for the weight loss, right? A lot of companies now, they try to follow this weight loss. You see, basically also winning your hands in the game of weight loss drugs. And I think that's really interesting research. And I'm looking forward to people following up on that. That would be great. At this point.
And I love the use of the word beautiful, right?
Yeah.
It's beautiful because these layers are all agreeing. It's giving us a preponderance of evidence that gives us a lot of confidence. That gives us confidence in the other results that you see too, right? So the fact that you've built this systematic map of these potential causalities, this corroborates, I think, the approach, which is certainly beautiful.
Great. At this point, I would like to thank Mine and Claudia. We can stay here and discuss for hours or for days with this amazing paper and amazing data. We learned so much today. And I would like to invite you, if you have to add something to our audience, Mine or Claudia, from your perspective and where you see this going to the future, that would be great to have your feedback and your thoughts about. Yeah,
anything you'd like to add, we'd love to hear it.
No, I think currently, as I mentioned, the increasing large-scale, high-throughput generation of different layers of biological data is currently very exciting, and I'm very excited to be in a field where we're better understanding their potential translational capacities. And with that, I also would like to thank both my colleagues, which have enabled all the work that we do possible, as well as the participants of Epic Norfolk study and the past and present team members, which have made the study possible.
Anything more from you, Claudia?
Yeah, I think I said already, moving forward, how it'd be very valuable to increase the kind of phenotypic space and the diseases we can look at. I think also moving forward, I certainly think, the beauty of these large-scale population-based studies is one thing, but certainly one of the reasons why I love sitting here in East London and being like just 100 meters away from one of the largest hospitals is because the next step really is how do we enable, clinical proteomics, i.e. proteomics in patient studies? How do we design that well and in a way that enables flexibility of different research questions? So that's kind of what I want to focus on, and which I think will be very exciting. And for that, we will need technologies that are kind of ready to move to that standard relatively quickly. And so I think that's an exciting new area, and doing that in a way that is affordable, even within the context of a national health system. So it's exciting. It's a really amazing times, and working together with talented people like Mina and other people in our team. It's just, as you say, and as actually one of my mentors say that, if it's not fun, it's not epidemiology.
I love that. Well, you certainly make it look fun, that's for sure.
Now, thank you so much for the opportunity to talk today.
It was our pleasure to have you.
Pleasure.
All right. Thanks, everyone.
Thank you.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.